Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on absorption of fats. Now, in the previous lecture, we had learned about the process of digestion of lipids. Now, when the lipid digestion is completed, the product which is formed, cholesterol is formed, monoglyceride, lysolecithin, free fatty acids are formed. And all this lipid digestion product, they solubilize in the intestinal lumen by the formation of mixed micelle. Now in the micelle formation, glycerol is not taking part because glycerol is water soluble, that's why. Now before we proceed in this topic, I would like to revise few concepts. First, the emulsification concept. In the figure, this is the large fat globule is shown, and the surface of this fat globule, we have learned that it is covered by lecithin and the bile acid, and these two, they are decreasing the surface tension. So the large fat globule is converted into smaller fat droplet, and this smaller fat droplet, they are known as the emulsification droplet. And as you can see on the surface of the emulsification droplet, so here the bile salt is present. These are the bile salt or the bile acid, and this is the these are the lysolecithin. This one is the core region where the lipids are present. Now, on the emulsification droplet, colipase. Colipase is a protein which is present in the pancreatic juice, and the function of this colipase is to remove this bile acid from the surface of the core lipid. Now, once the bile acid are removed, these enzymes, pancreatic lipase, cholesterol ester hydrolase, and phospholipase A2, they act on this core lipid. And we know the lipid, mainly the dietary lipid, they are in the form of triglyceride, and in the structure of triglyceride there is one glycerol molecule and there are three fatty acid, and these three enzyme, they are breaking the ester bond. So when the ester bond are broken down, so monoglyceride is formed, fatty acids are released. So this is the process of lipid digestion, and we get monoglyceride, cholesterol, fatty acid, fat soluble vitamins, and lysolecithin due to the process of lipid digestion. So these are the product of lipid digestion, and this product of lipid digestion, they participate in the micelle formation. Now, what are the micelle and how the micelles are formed? Okay, so this was the first point, first theory point. Moving on the second point. Now, micelle, the mixed micelle are cylindrical shape disc, and the average diameter of the micelle 50 angstrom. Now the core, that means in the center of the micelle, the lipid digestion product are present, and this lipid digestion product, they are covered exteriorly by the bile salt, and the bile salt are the empathic. So here in the figure, one missile is shown, and what is the missile, how the missile is formed. So these are, if you see, these are the lipid digestion product. This is CHOL, that means cholesterol, Mg, monoglyceride, and the key is written over here. For example, LYSOPL, that means lysolecithin, and FFA means free fatty acids. So these are all lipid digestion product. And where this lipid digestion product are present? So they are in the lumen of intestine. And we know in the lumen the bile is poured, and in the bile there are the bile salt. So the bile salt, they are present around this lipid digestion product, and this is how the missile is formed. So missile is cylindrical disc shape. And what is the meaning of empathic? Bile salt are empathic. What is the meaning of this thing? So let's understand this point. Now first, the composition of bile, what the bile is made up of. So in the hepatic bile, 97% water, 0.7% bile salt, 0.2% bilirubin, 0.51% fats, cholesterol, fatty acid, and lecithin. So this lecithin is present in the bile. So here two main factor for this discussion: bile salt. So in the bile salt and lecithin. Okay. Now the typical bile salt structure of the bile salt is shown over here. This part, blue color part, this is the polar hydrophilic portion. That means this part is attracted to water. Okay, that's why it is hydrophilic portion. And this part, this the tail part, is non-polar hydrophobic portion. Now once again, in this figure I am explaining how the missile is formed. See, in the core, that means in the center, in the middle, free fatty acids are there. Fatty acids are produced, fatty acid are released by lipid digestion. There is a formation of monoglyceride, then cholesterol, then fat soluble vitamins are there, and all this lipid digestion product, they are covered by bile salt and the purple color molecule. So these are the lecithin. So lipid digestion product, they are covered exteriorly by the bile salt and the lecithin. Now in the structure of bile salt, you see this part, this is the polar side, and polar side chains are the hydrophilic side. That means this side, this part associate with the water, and this surface is the hydrophobic side, and this surface attaches to the lipids. Okay, so here you see, here all this inside the yellow color portion is the digested lipid product, and you see this, the hydrophobic surface is attaches, it combines with the digested lipid product, that are the fatty acid, cholesterol, monoglyceride, then the lysolecithin also over here, and one more thing, fat soluble vitamins. Okay, and these are the exterior lecithin molecules. So this entire structure which is cylindrical disc shape, this is known as the missile. Okay, so these missils are formed in the intestinal lumen. So this figure is important for you, so I am showing you the entire figure this way. All the information you go through. Okay, now here this thing we need to understand, what is this. So here in the beaker there is a water. Now in this beaker, which is half filled with water, we are adding the oil. After the addition of oil, we know oil will remain on the surface of water, and oil is not mixing with the water. Now in this beaker we are adding lecithin. So when we add lecithin into this oil water, what happens? The entire oil is converted into the small oil droplets. So these are the small oil droplet, and on the surface of this oil droplet the lecithin is present, and this lecithin is shown by the green color. Now this structure means the oil droplet covered by lecithin. We are zooming this structure. So inside, this is the small oil drop, and around this small oil drop, this green, these are the lecithin molecules. So if we observe this closely, this is the green color, this is the head part, and this black color is the tail, head and tail. Now again we are zooming this. So this is the structure of lecithin, and this green color part, plus minus, that means polar region. This is the polar head, and this head is the hydrophilic, means this head is towards the water, and this part, purple color part, is hydrophobic. So, and that means it is non-polar and it is away from the water, and this tail, this hydrophobic tail, is attaches to the oil surface. Now in the missile formation, we can understand, instead of oil, there are the lipid digestion product, and around this lipid digestion product you see the arrangement of the head and tail. So the head is hydrophilic, and the head is towards the aquatic phase. Aquatic phase meaning uh, we know in the aquatic phase means the lumen of intestine, there is the intestinal juice and the pancreatic juices are present, and in this juices the water is there. So the head is towards the aquatic phase. And the bile salt are amphipathic, means they are also having the hydrophilic and hydrophobic region. Okay, that is the meaning of amphipathic bile salt. So this is how the micelles are formed in the intestinal lumen. Now what happens to this micelle, where they are going? These micelles are. So this point I have explained to you that this is the fourth point. Hydrophilic portion of the bile salt molecule, they are dissolved in the aqueous solution, that means in the water of the juices of intestinal lumen, and this is how these bile salt are solubilizing the lipids by forming the micelle. Okay, now the fifth point. Now fifth point is written over here. You can read this. I am directly explaining you. Now here what happened, this cell, this is the cell, this is the enterocyte cell, epithelial cell of the small intestine, and here these are the microvilli. These microvilli together, they are forming, they look like, this all microvilli together they look like the bristle of brush. So that's why this border is known as the brush border. So in the lumen of intestine, these micelles, they are migrated, they are reaching towards this brush border. And for simplicity, I can tell you that the micelles are emptying. So all the product of lipid digestion are entering into the enterocyte cell. Now, which are the lipid digestion product and which lipids are entering into the enterocyte cell? Now here you see what is happening. Cholesterol is entering into the enterocyte cell, monoglyceride are entering into the enterocyte cell, then lecithin and free fatty acid, all these are entering into the enterocyte cell of the small intestine. So we are returning back to that figure that I was showing to you, this figure. Okay, so here the product, lipid digestion product, they are diffusing, they are entering into the enterocyte cell along the concentration gradient from high to low, from lumen to cytoplasm of enterocyte cell. Moving on the sixth point. Now before I discuss this sixth point, I would like to show you this figure. This figure is also very important one and good one. Now this is the emulsion droplet, small droplet of lipid, and now we know there are three enzymes which are involved in the digestion of lipid. So lipids are digested and monoglyceride is formed, and from this droplet along with monoglyceride long chain fatty acids are released, short chain fatty acids are released. Now, this released monoglyceride, released short chain, long chain fatty acid, all this are covered by the bile salt. So in the core region, what is there? In the core region, there is the short chain, long chain fatty acid, monoglyceride is there, cholesterol is there, lecithin is there, fat soluble vitamins are there, and all this, they are exteriorly, they are covered by the bile salt. And now the structure which is formed by the combination of this lipid digested product and the bile salt, this structure is known as mixed micelle. Now what happens to this mixed micelle? Where is this mixed micelle right now? So this entire region is the lumen of small intestine, and in the lumen the mixed micelle are present. And this cell this cell this cell is the this cell is the enterocyte cell of small intestine. Now here, this one is the brush border of the enterocyte cell, and you see, in the brush border, this one is the sodium hydrogen exchanger. And what this exchanger is doing? So this exchanger is exchanging Na plus ion and H plus ion, that means the protons. So the protons are coming out, and the Na plus, they are entering into the cytoplasm of the enterocyte cell. So as the H plus are coming out, this lumen become acidic, and this acidic environment is favoring the lipid product to be released out from the micelle, and this lipid digestion product, they can now enter into the cytoplasm of the enterocyte cell. Now for the entry of the lipid digestion product, there are three mechanisms. First mechanism is the non-ionic diffusion. So here, number one, lipid digestion product, let's say the fatty acid, and the fatty acid is entering, okay, by the non-ionic diffusion mechanism. Second mechanism is the incorporation into the enterocyte membrane. So this one is the membrane, this is the membrane, and this one, number two, lipid digestion product. So this membrane is covering, okay, this membrane will cover this. So one vesicle will be formed, and in that vesicle the lipid digestion product is present, and the remaining, once this enter into this cytoplasm, what happen? This membrane again fuse together. So this mechanism is known as the incorporation into the enterocyte membrane. And the third is the carrier mediated transport. So here, this one is the lipid digestion product, and this is the carrier. So with the help of carrier, the lipid digestion product entering into the cytoplasm of the enterocyte cell. So since this figure is important, I am showing you the entire figure. Okay, this is the information regarding the figure. Now the sixth point. Now the micelles per se do not enter the cell. Micelles are not entering into the cell. If you observe this figure closely, only the lipid digestion product are entering into the cytoplasm of enterocyte cell. Now what happens to this bile salt? So here the bile salt are shown, and where this bile salt are going. So bile salt, they are forming the bile salt micelle. And what happened to this micelle? Now the bile salt, they are left behind in the intestinal lumen, and then they are absorbed downstream in the ileum. So in the ileum the bile salt are absorbed. Now the most ingested lipid, the lipid that which is present in our food, the dietary lipid, after the digestion, that lipid is absorbed mostly into the mid jejunum region. Now the work of the bile salt is completed. So now the bile salt, they return to the liver via enterohepatic circulation. So bile is returning to the liver via enterohepatic circulation. Now moving on the seventh point. Now inside you can read this point, and I will directly explaining what is happening. So in short, this is the enterocyte cell, and in this enterocyte cell again the lipid formation is taking place. What is happening? I'm showing you in the figure. This figure, this is also a very nice figure. Now you see what is happening in this figure, that see, cholesterol, from the micelle cholesterol is entering, monoglyceride is entering, lecithin entering, and free fatty acid are also entering. Now with the cholesterol, free fatty acid combine, and CHOLE, that means cholesterol ester is formed in the enterocyte cell. Then monoglyceride combined with free fatty acid, triglyceride is formed. Lecithin, free fatty acid, PL, phospholipids are formed. Okay, so this is how the digested lipid enter into the enterocyte cell, and this is how the lipids are once again they are formed in the enterocyte cell. So we are moving back to our discussion. So seventh point we were discussing, and this is the meaning of the seventh point. Okay, they have written over here. So lipid formation in the enterocyte cell. Now what happens to this newly synthesized lipids? So inside the cell, the re-esterified means, let's say we have monoglyceride. In the structure of monoglyceride, we know there is a glycerol and fatty acid, and between the glycerol and fatty acid there is a ester bond. And if we add two more fatty acid to this monoglyceride, so this is the new ester bond, this one is the new ester bond. So that's why this new lipid, we can say this lipid are esterified lipid. Now, this newly synthesized lipid in the cytoplasm of the enterocyte cell, they are packaged with the apoproteins. And how this packaging is taking place in the cytoplasm? So, this lipid digestion product from which the new lipids are synthesized, they combine with the apoprotein and lipid carrying particles are formed, and this lipid carrying particle are known as the chylomicron. So with the figure I will explain you the structure of chylomicron. So when you look into the figure, then you will understand what are the chylomicron and how they are formed. But here we let's finish this point. Now the chylomicron, the diameter, average diameter of chylomicron, that is 1000 angstrom, and this chylomicron, they are composed of triglyceride and cholesterol at the corner, that means in the center or in the middle part. The triglyceride and the cholesterol are present, and on the surface phospholipids and the apoprotein are present. So I am showing you this arrangement in the figure. The same figure, see. So these are the newly synthesized lipid. Okay, now in the center cholesterol ester, so this is the cholesterol ester. So in the core region cholesterol ester and TG, triglyceride, are present, and on the surface PL. So on the surface phospholipids are present, and on the surface apoprotein, ApoB, is also present. So this new synthesized particle in the cytoplasm of enterocyte cell, this particle is known as the chylomicron. So once again we are moving back, and if you see this chylomicron, only 20% portion is covered by this apoprotein, and rest of the 80% area is covered by phospholipids. So once again we are coming back to our main discussion, returning to the main discussion about the chylomicrons. Now what happens to this chylomicron? Where these chylomicrons are going? How they are leaving the enterocyte cell? That is the question. So here, this point I have explained you. Okay, 9 point. Now the 10th point. Now the apoproteins, they are synthesized in the intestinal epithelial cell, and this apoprotein, they are essential for the absorption of chylomicrons. Now the person who is not able to synthesize this apoprotein, let's say the ApoB or the beta lipoprotein, so the person suffers from the disease known as beta lipoproteinemia. And in this condition, in this situation, a person is unable to absorb the chylomicron, and That's why the person suffering from this condition is not able to absorb the dietary lipids. Now, returning back to the main discussion of the chylomicron. Now, the theory point is written over here. Now, here, this one is the chylomicron. Now, in the cell, what is happening? So, see, the chylomicron, where they are going. So, chylomicron are packaged into the secretory vesicle on the Golgi apparatus. So, this chylomicron, first of all, it will reach to the Golgi body. It will fuse with the Golgi body, and the Golgi apparatus packages this chylomicron. So Golgi apparatus releasing the secretory vesicle, and in the secretory vesicle, you see over here the Golgi apparatus is shown, and the Golgi apparatus is releasing this secretory vesicle, and this secretory vesicle are having the inside chylomicron. Now this secretory vesicle, where they are reaching, where they are going. Now this is the basolateral membrane. So this secretory vesicle, they reach to the basolateral membrane, and here this secretory vesicle burst, and since the secretory vesicle are bursting over here, what happened? The chylomicron present inside, they are released. Now where this released chylomicron are going? That is the question. So now, last point of this lecture, point number 12. Okay, now the point number 12 is written over here. Okay, now the chylomicron are too large to enter the vascular capillaries, but the chylomicron, they can enter into the lymphatic capillaries, and these lymphatic capillaries are known as the lacteals. Now I know this is a bit difficult, so with the help of the figure I am explaining this last point, so you will easily understand that. Now this figure, what is this? This one is the villus of small intestine. Okay, in the villus, what is there? Blood capillary there, lacteal is there, arteriole is there. Now in the green color lacteal, they have already shown the chylomicron, but the question is how the chylomicron entered into the lacteal. That is the question. Now here, this entire structure is the villus. This is the villus. Okay, and this part is the villi. Okay, and I am zooming this villi. So when I zoom this, you can understand over here the enterocyte cell are present. Okay, and this cell here, this one is the enterocyte, then over here enterocyte cell, enterocyte, and this is the breast border. Okay, and now one secretory vesicle here, the secretory vesicle bursting, chylomicron, these are the chylomicron are releasing, and where this chylomicron are going, so they are entering into the lacteal. Okay, now in the lacteal what is there? So lymph is there. Okay, now this chylomicron, then this lacteal. Now what is happening to the lacteal? So lacteal is merging with lymphatic vessel, then this lymphatic vessel merge with thoracic duct, and thoracic duct is pouring the lymph to the main blood circulation. Okay, so this is how the chylomicron enter into the main blood circulation of our body. So this is the 12th point, and one figure I have shown you, and for the 12th point I am showing you one more figure, this one. Enterocyte cell is shown, then chylomicron, how they are released, where they are going, this is the information. Then absorption, concept map of absorption for carbohydrate, protein and the lipid. Then this figure, concept map for the absorption of carbohydrate, protein and fats. Okay, this way, this is the half part of the figure. So with this we have completed the discussion of absorption of fats. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kushti sir. I am from Ahmedabad, India. Bye bye. Namaste.